In this video, we will discuss about the pro-aging potential of aged CD8 plus T cells. So in the previous video, we have understood how these aged CD8 plus T cells and the young CD8 plus T cells behave differently based on the environment in which they are present. And now researchers wanted to check whether these aged CD8 plus T cells have the potential to accelerate aging or not. So for that, what they did is they used this hippocampus as the experimental model, and in that they injected young CD8 plus T cells and the aged CD8 plus T cells separately. That is, in two different hippocampus they used two different injections, and they also have some replicates. So after that they did the analysis in the young mice, and after 24 days they analyzed the cells, and they analyze it using bulk RNA-seq data. So in that bulk RNA-seq data, they are able to see that the aged CD8 plus T cells accumulate more towards this neuron and this brain barrier, whereas in the case of young CD8 plus T cells injection, they do not observe that much accumulations near the neuron and brain barrier, indicating this aged CD8 plus T cells might have some role on the function of this brain. So thereafter they did this bulk RNA-seq. They used the bulk RNA-seq data and also the single nucleus RNA-seq data of hippocampus to find out what are the different genes that are dysregulated or differentially expressed in the aged CD8 plus T cells and the young CD8 plus T cells, and they are able to find out that the list of the genes, some of the list of the genes using shown in this heat map, which are overexpressed in the case of young CD8 plus T cells but are downregulated in the case of aged CD8 plus T cells, and these are related to some of the functions that are mentioned here. That is, mainly it is related to this regulation of synaptic activity. And when this dysregulated expressed genes are analyzed using the gene ontology analysis, they are able to see that this genes, the pathways that is differentially regulated or upregulated in the aged CD8 plus T cells compared to the young CD8 plus T cells is mainly this translation at synapses. That is, it will increase the synapses translation. Whereas when we check the downregulated in the aged CD8 plus T cells compared to the young CD8 plus T cells, is this regulation of this membrane organization, basement membrane organization. That is, the membrane organizations is usually downregulated. This could means that the cells might face problem in this during this membrane reorganizations, and if we combine all this towards this brain, one can see that the results of this bulk RNA-seq and single cell nucleus RNA-seq data using this gene ontology analysis shows the relationship with the synaptic and which might affect this brain functions in the case of aged CD8 plus T cells. And they have in the panel A, they have shown the experimental system where they have checked all other the physical activities, which has those results are not shown in this figure, but we will discuss the further results in this figure or in the subsequent videos. So I hope you understood how they plan to check their hypothesis about the pro-aging potential of aged CD8 plus T cells, and the results that are shown here. In the case of the aged CD8 plus T cells, they not only just accumulated in the neuron and brain barrier, but they also accumulated at other places. You can check the text to find out the detailed insight, and also you can also refer to the supplemental documents that also shows this aged CD8 plus T cells accumulations at different places. So I hope you understood the results that are presented in this figure to some extent. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.